Okay, so I realized that I did not make a video for those of you who were unable to attend class. So let me just do that now so that we can get you guys all up to date on the lecture material that we talked about in class. So, okay, let me go back to here. Okay, so what we're going to do is we're going to talk about genetics and inheritance now. And we're going to link these concepts back to what we learned with meiosis. All right, so just to review, we have a gene, which is our unit of heredity. It's being transferred from parent to offspring. This is a distinct set of nucleotides, so it's a specific sequence of A, C's, G's, and T's found in our DNA. These code for proteins. We talked about that at length. And then humans in our genomes, we have somewhere between 20,000 and 25,000 genes. Genomes is all of the genes that we have in a singular organism. And in diploids, most of the time chromosomes come in pairs. And so here I threw up this image of a karyotype of some organism that has 18 chromosomes. So it's definitely not a human. Okay, so remember, a karyotype is just a figure that shows all the chromosomes of the critter. Okay, let's talk about Mendelian genetics. Love Mendelian genetics. This is so exciting. Okay, so pre-Gregor Mendel, what we understood about heredity was that it occurs within a species, that we have, that there are some traits in the offspring that are similar to the parents. Like we can see there's characteristics of mom and dad in the offspring. But I love how like horribly vague this is. Traits were thought to be born through fluid and blended in the offspring. What the hell does that even mean? Well, it's pretty vague. So, but here's the thing. If this is correct, then why don't all individuals look alike? If we just see this blending of characteristics, like wouldn't we all eventually kind of like wash out to all look the same? Like if you think about throwing a smoothie together, you put all your strawberries and your raspberries and your whatever, or your banana or whatever, into a blender, and you go, and if you blended it well enough, then you shouldn't be able to see specific chunks of the thing. And so kind of shouldn't that be how our traits are? If it's this blending of traits being cast through fluid. So we're like, okay, that's probably not right. This is a page from the book called, let's see if I can find it quickly. The Cartoon Guide to Genetics. I have a PDF version of this if you guys are interested in this. It's a little bit dated, but it definitely goes through all of the concepts that we're going to talk about in class in a kind of more interesting and entertaining kind of way. But still gives you the information that you need. All right, so if you want that, let me know. All right, this Gregor Mendel words is a link to a TED Ed video. It's about 3 minutes long. I definitely highly recommend watching that, especially if you need a little bit of a refresher on Gregor Mendel. But this dude was a monk in I think Austria around the same time as Darwin. So Gregor Mendel definitely knew about Charles Darwin, although Darwin didn't know about Mendel. So Gregor Mendel was interested in understanding how these traits are passed on from parent to offspring. And the model organism that he used are pea plants. And pea plants are great because they're able to hybridize and create viable offspring that we can then go on to use for future generations. There's lots of different kinds of peas, so you can find lots of different varieties. So all of your peas are going to be able to do different things, have different traits. Peas are small, they're easy to grow, they mature quickly, so you can probably get 2 generations within a single growing season. So that's handy for just the logistics of doing science. And then this one is a big deal. So peas can self-fertilize or be cross-fertilized. So what he would do is he would pull the male parts of the plant off and then intentionally give it, place the pollen on the female of the same flower or of a different plant, depending on if he wanted to do a self-cross or a cross-fertilization. Okay, so that's Gregor Mendel. All right, so let's remember some of these words, and these words are actually on your meiosis lab sheet. So the genotype of a critter is all the alleles, all the genes, all what the type of genes that this critter has, right? So you can think of genotype as the type of genes, all the alleles contained within this individual. Now, let's remember what an allele is. Well, these are different copies of the same gene. So in Gregor Mendel's case, he was looking at pea plants that have purple flowers or white flowers. Each of these things are describing the color of the flowers, but they're different colors. So those are different versions of the same trait. And so we can talk about round peas and smooth peas, or yellow peas and green peas. Or so, you know, these are the same traits, different versions of that trait. If you have two different alleles on your homologous chromosomes, then that means that you're a heterozygote. So hetero means different. So you have different alleles. If you're homozygous, that means you have the same allele. And you can either be homozygous dominant, having two dominant alleles, or you can be homozygous recessive, having two recessive alleles. But you cannot be heterozygous dominant or heterozygous recessive, because that's silliness, because if you're heterozygous, you have different things. So you can't be dominant or recessive with heterozygous. Okay. All right. Okay, so the phenotype then, so here we go, some more vocab for you, is the physical or measurable characteristics of an organism. And so I put my puppies up here mostly because I wanted to put a picture of my dogs on the slides, but I wanted to show you the differences of their phenotype. So Maggie has two different color eyes. Sadie has the same color eyes. Sadie has a dirt face. Maggie is very curious, and she's a goofball. She's the class clown of all the dogs that live in my house. Sadie's the protector. So they have very different personalities, and they look different. Obviously, their fur's color is different. You can tell that they're sisters because there's similarities in their phenotype, but there definitely are significant differences too. Okay, so in us diploid organisms, just because we have a recessive allele, I should probably tell you what dominant and recessive allele is. So if you have one or two dominant alleles, then that trait is the trait that is expressed. For example, so I'm left-handed, but all of my kids are right-handed. So I have two recessive alleles for handedness, meaning that I am left-handed. All of my kids got a recessive allele from me, but they all got a dominant allele from their father, because, and we know this because all of my kids are right-handed. Okay, so with that in mind, think about this question. If you see a dominant phenotype, can you tell if the genotype is heterozygous or homozygous dominant? Can you tell just by looking at a creature? Probably not. But there's a way that you can tell, and I kind of described it. So one of the ways that you can describe it is by using mating. And so in my case, right, like I'm left-handed, that's a recessive trait. I have two recessive alleles. I mated with somebody, and at best, my kids can be, my kids have to be heterozygous. So if we know the parents, the genotype of the parents, we can kind of deduce the phenotype or the genotype of the kids. Or if we wanted to know, like, okay, so anyway, if we see that the recessive phenotype, then we know that the genotype of that individual. Being I'm left-handed, so that means that we know my genotype as far as handedness goes because it's recessive. Okay, let's move on. All right, so let's just talk a little bit more. I'm going to put me down over here so I'm out of the way. This figure. Okay, so the way that we can figure this out is by doing monohybrid crosses. So these are looking at the variability of a single trait. So we're going to look at two variations of one trait. And so that's where the mono comes from. Mono means one, and so we're looking at a single trait in these crosses. So let's look at the case of Gregor Mendel's peas. So we have some pea plants that are true breeding. And what true breeding means that if you cross this with itself, you're going to get purple flowered offspring every single time. And then if you're going to cross this guy, this true breeding white flowered variety, you're going to get white flowers every single time. Okay, and so one of the ways that people use this concept now is we call them heirloom varieties. So if you buy some heirloom tomatoes, what you can do is take that seed out of that plant that the fruit that you get at the grocery store, and if you planted that, you would get tomatoes with the same traits as their parent. And so now when you get like spaghetti sauce, and they're like made with heirloom tomatoes and we're gonna double the price because they're fancy, you can just be like, yeah, hello, you're just homozygote dominant. Like, that's what heirloom means. Okay, so let's talk about this. So if you have these parental plants and you cross them with each other, all of their kids are going to have violet flowers or purple flowers. So, okay, great. So now we're going to take that. So all of this F1 generation, this first generation, is going to have all of the dominant traits. And then if we take their seeds or those flowers and we cross them with themselves, then in the second generation, we start to see that 3 of these individuals will have on average, if we did the math properly, we would see that about 3/4 of the offspring would have purple flowers and about 1/4 would have white flowers. And so even though this recessive allele went away in that first generation, it popped back up in the second generation, in the grandkids. So, that's interesting. How'd that happen? Okay, so what Gregor Mendel, he's found, and so he looked at a lot of different traits: flower color, seed coat texture, plant height, seed pod color, seed, all the things, right? Like all these different traits. And he kept finding this 3-to-1 pattern show up in the second generation. That's crazy. I wonder what's up with that. And so he was actually able to use mathematical models to predict what the genotypes might be. So he took some plants that he didn't know the genotype of and he crossed them. And if he got this 3-to-1 ratio on the second generation, he could say, this is evidence that this grandparent genotype is homozygous in both plants. Okay, so, and this is now why, so we can thank Gregor Mendel for having to do statistics and rubbing math on stuff in order to turn it into science. So we now have mathematical models because of Gregor Mendel. All right. So if we take each of these individuals from our second generation and we cross them with themselves, what we see in the third generation, so generation 1, generation 2, generation 3, is that we see a 1 to 2 to 1 genotypic ratio. So we see that These guys are all going to have purple kids. All these guys are going to have all white kids. And then there's going to be some genotypic differences there. Okay. So what is the phenotype ratio of this generation? So what we can do is we can count up. Okay, in this example, we have 1, 2, 3, 4, 5. We have 16 total. Okay. Ooh, I don't know if you can see that. Okay, we have 16 total. We have, let's see, how many purples do we have? We have 1, 2, 3, 4, 5, 6, 7, 8, 9. Did I count that right? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Nope. Okay. And then that means that we have white. We should have 6. 1, 2, 3, 4, 5, 6. Great. Okay. So now in order to get the ratio, we first need to get, so what we're going to do is we're going to take 10 of our purple guys and divide it by our total. And then that's going to give us some number. And then if we did that for the white ones, that's going to give us some number. And then we can reduce these down in order to make a ratio that's similar to this. Okay. And so what I used to do with my, when I taught 181 at NAU, is I would give these kids a corn cob and that had all these different colors. And so then each of those seeds would be resulting of different pollen. And so then they would have to count up how many purple ones they saw and how many yellow ones they saw and how many white ones they saw. And then they would have to use these mathematical models to figure out what the genotype and the phenotype ratios of that. I won't make you guys do that, but I could. Okay. All right, so now here's the question. How does these principles of inheritance fit with what we learned in meiosis? So maybe this is going to be a particularly good essay question that I'm like, okay, link these two things together. We have Mendelian genetics where we see traits that disappear at the F1 generation and then reappear in the F2 generation. And so now you can start to think about, okay, well, during meiosis, we have these two sources of variation, genetic variation. One's in crossing over, the other one is an independent assortment. Okay, so now, okay, so if we have these shufflings of these genes, one of those alleles might be dominant, one might be recessive. Okay, and so we might see these 3-to-1 patterns show up due to independent assortment. Okay, so you could say something around those lines. We're going to skip this slide for today. And now what we need to do is talk about Punnett squares, because these are very powerful tools made by a dude called Reginald Punnett. And he wrote the first genetics textbook back in 1905 before we, pretty much based on Mendelian genetics and what we knew at that point. So let me, sorry, I got to grab an eraser because I want to draw out some Punnett squares on the board here. So what I'm going to do is actually stop sharing my screen so that you guys can see my whiteboard better. Okay. So Punnett squares allow us to get the predicted phenotype and genotype ratios of some offspring. So let's say, for example, come on, autofocus. Okay, let's say, for example, that we have a, our dad gametes are going to be, let's say, let's make them Q's because those are easier just to tell the difference between uppercase and lowercase when I'm handwriting. Okay. So those are going to be, so our dad is going to be heterozygous in some trait. Let's choose a trait. Let's say loudness of voice. I don't know. because I was like, what starts with a Q? Quiet. All right, sure. Okay, so our dad's gonna have that, and then our mom, let's make our mom also be heterozygous. Okay, so mom and dad are both really quiet. We'll say that the capital letter is our dominant allele, our lowercase letters are recessive allele. So if you have the dominant allele, then you're quiet. If you have two recessive alleles, then you're loud. I don't know, whatever. Okay, so let's draw ourselves our little Punnett square here. So the types of gametes that dad can make are a big Q and a little q. The types of gametes that a mom can make are a big Q and a little q. And the way that we do this is now all we have to do is just bring these down. So dad can give us a big Q for this potential combination. Mom can give us a big Q for this potential combination. Dad can give us a little q, and mom can give us a big Q. Oh my God, that looks weird. Okay. Whew. Okay, that looks better. Big ones, just kind of by convention, the big ones usually go first. All right, so then we're going to take our big Q from dad and our little q for mom, and then we're going to take our little q from dad and our little q from mom. Okay, now come on, out of focus. Yeah. Does this give us the, so if this mom and dad had 4 kids, are we guaranteed that one of the kids is going to be homozygous dominant, 2 are going to be heterozygous, and 1 is going to be homozygous recessive? No, this is the probability that each of those things are going to happen. Each time that we see this recombination of gametes, these are independent possibilities. And so let's talk about what that means with, let me go back to sharing my screen. Okay. Okay, let's talk about the rule of multiplication and probability. So, okay, so here we go. Rule of multiplication is the probability of 2 independent events conferring simultaneously is the product of their individual probabilities. What the hell does that mean? Okay, let me find my dice. Where did I put them? They're over here. So this is some of my dice. I love dice. You guys know I'm nerds. It's fine. Okay, so let me talk about independent events. So if I rolled this dice, I got a 6. Does the result of rolling this dice influence the results of rolling this dice? They don't, right? These are independent. I can roll this dice and I have a 1 in 6 chance of getting a 6. I can roll this dice and get a 1 in 6 chance of getting a 6. I can roll any dice. Every time I roll a dice, I have a 1 in 6 chance of getting a 6. Or pick your favorite number from 1 to 6 in this case, okay? That means that these are independent. They don't depend on each other. Now, so just to kind of give you an example of the contrast, let's say that your height might depend on your gender. So on average, men tend to be taller than women on average. So if we wanted to look at height, we would also want to be recording gender because those two things kind of do vary a little bit. Those aren't really independent of each other. They kind of depend on each other a little bit. Okay, so but these dice roll, coin flips, gamete combinations are independent. Okay. And so if I want to, let me flip here. Okay, so the probability of me rolling a 6 on this dice is 1 in 6. So let me erase this board now so that we can do some maths. Okay, so for my black dice, my probability of rolling a 6 is 1 in 6. Okay, so now what's the probability of me rolling a 6 on my red dice? Well, it is also 1 in 6. Now, what is the probability that I'm going to roll 2 6s on both of these dice when I roll them at the same time? Okay, this is these 2 independent things happening simultaneously. Okay, so in order to get that, what I need to do is all I got to do is multiply. Now, if you can remember way back to like elementary school, the way that we multiply fractions is we multiply the numerators. 1 times 1 is 1, 6 times, and then we multiply the denominators. 6 times 6 is 36. So the probability that I'm going to roll both 6s on these dice right now. Ooh, I just rolled 2 5s. But I was trying to roll 2 6s, so that doesn't count. Okay, so okay, I've lost my train of thought. Oop, okay, let's talk about probability. So here we go. This is basically what I just said, but with pennies, and this kind of looks like a Punnett square, doesn't it? So if we have a penny, the probability that we're going to flip this coin, and the first result that we're going to land on heads is about half, and the probability that it's going to land on tails is about half. There's some very small probability that it's going to land on its side, but that's pretty much 0, so we're going to ignore it. Okay. So then the probability that we on our second toss that we're going to get a head is about half, and the probability that we're going to get tails is about half. And so the probability that we're going to get 2 heads on our first toss, oops, is 1/2 times 1/2, which is equal to 1/4. Okay, and so we could do that for all of these things. And this is basically the same as what we just drew with Punnett squares. So I could ask you a question. What is the probability if I have 3 6-sided dice? What is the probability that I'm going to roll all ones? And then I would ask you to calculate that probability. And you could probably do that now. Okay. I'm going to stop here because these dihybrid crosses are more complicated and warrant their own video. So look for that in just a minute.